Ozempic was supposed to reduce hunger, but researchers started noticing something stranger. Some people weren't just eating less. They were losing interest in things they had wanted for years. And that discovery led scientists to a hidden system in the brain that decides what to desire.
Part 1. The woman who forgot to want. She opens the refrigerator, stands there for a moment, and walks away. Not because the food looks unappetizing, not because she has already eaten. She reaches the cabinet where she has kept the same snacks for years, the ones that have occupied her thoughts every evening, and realizes she simply is not interested. There was no decision, no willpower, no moment of discipline. The pull was simply not there. This is the detail patients consistently struggle to explain. Not that they ate less, that they wanted less. The distinction seems minor. Biologically, it is enormous. Semaglutide, marketed as Ozempic, Wegovy, belongs to a class of medications called GLP-1 receptor agonists. Originally developed for type 2 diabetes, it became globally known for producing substantial weight loss. The mechanism appeared obvious: mimic the body's natural satiety hormone. Feel fuller sooner, eat less, lose weight. That explanation was accurate. It was not complete. Because hunger and desire are not the same biological process. Hunger measures need. The body registers an energy deficit and signals it. Desire measures motivational priority. The brain determines how strongly that need should be pursued, how much of its limited behavioral resource it is worth spending. Normally, these two systems operate in such close synchrony that they feel like a single experience. You are hungry, and you want to eat. The need and the drive feel inseparable. What patients were describing suggested those two systems were separating. The body's need for energy remained. The brain's drive to pursue it had quietly diminished. The investigation shifted away from the stomach. Attention started moving toward the brain. Part 2. The brain was already listening. What researchers discovered was not what they expected to find. GLP-1 receptors, the exact molecular targets semaglutide activates, are not confined to the digestive system. They are present in the brain, specifically in structures that have nothing to do with digestion. The nucleus accumbens, the ventral tegmental area, the hypothalamus, the prefrontal cortex. These are not metabolic regions. These are the structures that govern reward processing, dopamine signaling, motivational evaluation, and the behavioral decisions that emerge from all three. Dopamine's role in this system has been consistently mischaracterized in popular accounts. Dopamine is not the pleasure chemical. The brain does not release dopamine because something feels good. It releases dopamine in anticipation of something predicted to be rewarding. Dopamine encodes expected value. It is the signal that motivates approach behavior before any reward is obtained. When a rewarding experience occurs, dopamine activity updates the system's estimate of that target's future value. The next time a related cue appears, dopamine fires in anticipation. The reward acquires pull. The brain begins treating it as something worth pursuing. That process, the encoding of expected reward, is how ordinary preferences become habits. How a glass of wine with dinner becomes something that feels necessary. How a nightly phone check becomes something the hands do before the mind decides. How doom scrolling at midnight becomes something the body moves toward without a conscious invitation. Semaglutide, by activating GLP-1 receptors across these regions, was not simply influencing satiety. It was entering the circuitry that determines what deserves pursuit. Researchers were beginning to understand they were looking at something larger. Beneath appetite, beneath satiety, beneath the visible metabolic effects, the brain contains a system that continuously assigns value to the world. A system that ranks competing targets, encodes their importance, and quietly determines what receives the resource of effort. A desire machine, running in the background of every conscious moment, and semaglutide had just entered it.
If you're fascinated by documentaries like this, subscribe to Pathology of Fear. New medical documentaries every week.
Part 3. the system that decides what matters. The reports did not stay limited to food. Consider what patients were describing. A man who had poured a drink every evening for 11 years opens a bottle, looks at it, and puts it back. Not because he decided to stop, because the decision never formed. The pull he had felt every day since his 30s simply wasn't there. A woman who had compulsively checked her phone throughout the night, reaching for it before she was fully awake, scrolling before her eyes had adjusted to the light, finds herself leaving it on the other side of the room, not through discipline. The urgency had quietly evaporated. A man who had spent years cycling through online shopping, filling carts he would abandon, returning packages he had already forgotten ordering, finds the compulsion has dissolved. The familiar itch is simply gone. Individual accounts are not scientific evidence.
But identical descriptions from geographically separated, demographically unrelated individuals demand biological explanation. The explanation exists in the overlap.
Food is one of the most evolutionarily reinforced rewards the brain encounters. The neural systems that evolve to pursue food do not exist in isolation from other motivational systems. They share infrastructure with the systems that govern pursuit of alcohol, nicotine, and other reinforcing stimuli. All of these converge on the mesolimbic dopamine pathway, the same pathway semaglutide appears to influence. The brain does not build entirely separate architectures for separate desires. It builds one machine that ranks them all. Food, alcohol, gambling, sex, shopping, the phone. Every craving, every compulsion, every quiet preference that shapes daily behavior competes for position within the same evaluative system. Research in animal models has found that GLP-1 receptor agonists reduce alcohol intake and decrease the motivational drive to self-administer alcohol. Early human studies have produced preliminary signals consistent with reduced alcohol interest among semaglutide users. This research is ongoing, but the biological logic is no longer speculative. GLP-1 receptors in reward circuits. Documented dopamine pathway involvement, a single molecular signal capable of influencing motivational priority across multiple reward domains simultaneously. Appetite suppression is not the primary story. It is the visible surface of a much deeper change. Part 4. The quiet recalibration. Here is what the semaglutide data reveals about the desire machine. It can be adjusted. The alcohol is available. The motivational weight assigned to it decreases. The opportunity is identical. The assigned importance is not. The phone is in your pocket. The urgency that used to pull your hand toward it simply doesn't arrive. The snack is in the cabinet you have opened every night for years. The thought that used to begin forming at 9 p.m. doesn't complete itself. The targets remain. The world is unchanged. What has shifted is the internal ranking of what those targets are worth. This is the part patients find genuinely difficult to explain. Because it does not feel like willpower. It does not feel like discipline. Those would require an act of will directed at something that is pulling. What they are describing is something stranger. The pull is simply not there. The long-term picture remains genuinely uncertain. Not dangerous by current evidence, incompletely understood. Reward systems adapt. They reorganize around persistent inputs. Modern neuroscience has found no evidence that semaglutide causes neurodegeneration, structural brain injury, or progressive neurological damage. Those claims are unsupported. The open questions involve function, not injury. How a biological signal behaves when it remains continuously active. Whether motivational changes remain stable, diminish, or evolve across years of exposure. What the scale of semaglutide use has created, unintentionally, is the largest natural experiment in motivational biology ever observed. Millions of reward systems. Years of documented exposure. Behavioral data generated in real-world conditions that laboratory science rarely accesses. Answers are accumulating. They are accumulating slowly. Part 5. The things you want aren't chosen. Most people experience desire as something personal, something arising from their history, their preferences, their character. It does not feel like a calculation. It feels like a self. But consider what desire actually is before it surfaces into experience. A cue enters awareness, a smell, a memory, a familiar context. Neural circuits retrieve prior reward history associated with that cue. A predicted value is generated. Competing motivational signals are compared. A weight is assigned. The craving that arrives is not the beginning of that process. It is the end of it. The calculation was already underway before conscious awareness arrived. The ranking was already done. What surfaces into experience as a want, as preference, as craving, as the sense that something deserves your attention, is the output of machinery you have never seen. Over time, the targets that consistently receive high ranking begin to shape behavior in ways that extend far beyond individual choices. High-ranked targets become habits. Habits become routines. Routines structure time. Structured time becomes the architecture of a life. The foods you eat every day, the drinks that accompany certain hours, the way your hand moves toward your phone before you've consciously decided to reach for it, the compulsive checking, the late-night snacking, the doom scrolling, the gambling, the shopping. All of it assembled by a system that was running the entire time without your supervision. Here is how that assembly works over time. A target receives a high motivational ranking. Behavior moves toward it. The movement is repeated. Repetition encodes the pathway more deeply. The next time the cue appears, the ranking is slightly higher, the pull slightly stronger. The behavior requires less deliberate decision to initiate. It begins to happen on its own. That is habit formation at the biological level. Not a character trait, not a choice. A ranking reinforced by repetition until it no longer requires conscious activation. When enough habits cluster around the same targets, the same substances, the same rituals, the same compulsive patterns, they stop feeling like separate behaviors. They begin to feel like a person. The drink you always have, the way you always unwind, the thing you always reach for, the thing you cannot imagine not doing. And gradually, the things that receive the highest ranking begin to compose something that feels like identity, not chosen consciously, assembled automatically, one high valuation signal at a time. Consider what that means in practice. A person who has spent 20 years reaching for a drink at the end of a difficult day does not experience that as a biological ranking. They experience it as who they are, the kind of person who unwinds this way. The ritual that marks the boundary between the working day and the rest of it. The thing that belongs to them. A person who has spent years checking their phone compulsively, in line, in conversation, in the dark before sleep, does not experience that as a motivational system firing. They experience it as restlessness, as personality, as the way their mind works. The biological machinery and the felt sense of self have merged completely. There is no seam visible from the inside. Which is precisely why what happened next was so disorienting. What semaglutide disrupted for many users was not just a craving, it was that accumulation. And when the drug shifted the outputs of the desire machine, the downstream effects reached further than anyone anticipated. Not just appetite, not just alcohol, but the routines those habits had built, the patterns those routines had calcified, the sense of self that had quietly formed around them. For many users, the most unsettling part was not the weight loss. It was the quiet. The absence of something they had spent years assuming was them. Not through willpower, not through insight, not through any act of the conscious self. Through biology. This is what the semaglutide data actually revealed. Not a better weight loss drug. Evidence that the things you believe define you, your cravings, your compulsions, your relentless pursuit of certain things and your indifference to others, may be outputs. Outputs of a biological ranking system, running continuously, assigning value, quietly determining what the brain considers worth wanting, before any choice is made, before any self arrives to make it. The desire machine was always there. Semaglutide simply made it visible. And what it revealed is not a flaw, not a malfunction, not a disease. It revealed the normal architecture of a human brain, continuously ranking, continuously assigning value, quietly deciding what deserves pursuit. The things you want may feel entirely personal, and in some meaningful sense, they are. They have been shaped by your history, your experiences, every reward signal your brain has ever encoded. But beneath every desire is a biological system that was assigning value long before awareness arrived. And science has only just begun to understand what happens when that system starts changing.
Pathology of Fear is a medical documentary series presenting clinically documented neurologic and pathological mechanisms for educational analysis.